Hi everyone, and welcome to the tutorial on cells and biomacromolecules. Today we're going to be looking at the various cell types as well as the compounds that are essential for life. So, first of all, with cell types, we have prokaryotic and eukaryotic. Prokaryotic cells have no membrane-bound organelles and they have a circular strand of DNA, right, which comprises of all the genes essential for their function. They are really small and are unicellular. Okay? Example would be bacteria. Eukaryotic cells have membrane-bound organelles. They're much larger. All right, they have their DNA stored in the nucleus. Okay, and it is single-stranded. All right, these organisms are usually from unicellular to multicellular, and they have more complex organizational structure, as in plants and animals. So, what are these cells made of? Well, the main atoms they're comprised of are carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. These atoms combine to form large molecules, and the interaction with these molecules is what we're looking at today, as well as their synthesis. Molecules can be either non-polar or polar. Alright, the best non-polar molecules that we look at are oils and fats and lipids. Alright, they are not attracted to water, so what we do is we term them hydrophobic. These hydrophobic molecules prefer to interact with one another and stay attached to themselves. Okay, they have a high affinity to each other. Where polar molecules have regions of positive and negative charge within the molecule itself. This means that these positive and negative charges on the one molecule can interact with other molecules, forming like attractions. Okay, these attractions are known as hydrophilic attractions. Okay, they're water loving because water is a polar molecule. So when we look at water, we look at the way it forms hydrogen bonds, or the way it sticks to itself. Okay, because each water molecule is able to form bonds with another water molecule, and these bonds are called hydrogen bonds, and they occur because an oxygen atom is a very electronegative atom. Now, simply this means that it pulls the electrons towards it. Okay, so the electrons paired between oxygen and hydrogen are pulled more towards the oxygen. This gives hydrogen a positive charge and oxygen a negative charge. This charge is then used to form bonds with other water molecules. Okay, it allows water to interact right through hydrogen bonding. So, when we want to talk about the biomacromolecules, there are four main types: carbohydrates, proteins, lipids, and nucleic acids. So we're going to look at them today. So lipids are made of carbon, hydrogen, oxygen. Okay, their subunits are fatty acids and glycerol. They've got comprised of a fatty acid chain and a glycerol head. Okay, they are insoluble and they are non-polar due to the large hydrocarbon regions. Okay, they are used in energy storage and structural of the cell membrane and chemical signaling. So here I've outlined the function of the various lipids and what they do. Also, lipids can be saturated and unsaturated. This means that the saturated molecule of a lipid means it has no double bonds in the hydrocarbon chain. As you can see down here, it is a very straight chain and it is solid at room temperature. Unsaturated lipids have double or triple bonds between the carbon atoms. Alright, and what this means is they form kinks. Now these kinks mean that the lipids cannot sit next to each other. They cannot compact very nicely, and what this means is they become a liquid at room temperature. Okay, so glycerol is one component of a lipid. Alright, glycerol, the OH groups attach to three fatty acid chains which bond, and this bond releases water. So this reaction between glycerol and fatty acids is called a condensation reaction, and this creates a triglyceride, right, a saturated triglyceride, which you can see down the bottom right. And per fat there is one water molecule released. So per triglyceride, 3 waters are released. Okay, through condensation reactions. Lipids are not considered real polymers. Okay, because when you get a lipid, lipids do not bond with other lipids. So triglycerides do not bond with one another, okay, like carbohydrates and nucleic acids do, and proteins do, alright, and amino acids. They do not bond with one another. They just share a like affinity and therefore stick to one another through this affinity. Phospholipids, so the major component of cell membranes, alright, is comprised of a hydrophobic tail. Okay, this tail points inwards towards one another, right, and a phosphohead, which is hydrophilic. Okay, this phosphohead is polar, right, and that means it can interact with water on the outside. Okay, so the membrane can bond with the water, right, but it also prevents the water from flowing through due to these fatty acid tails, which are hydrophobic. Now, carbohydrates contain the exact same atoms as lipids do, okay, carbon, hydrogen, and oxygen, okay, but they occur in a ratio of 1 carbon to 2 hydrogen, 2 oxygen. The subunits of carbohydrates are monosaccharides, okay, and when 2 monosaccharides join together, you get a disaccharide. Now, depending on their size, okay, they are soluble in water, usually, okay, and they can't pass through the plasma membrane, right? They need channels in order to do this. Their very important cellular functions include chemical storage of energy. All right, they make up other compounds. So DNA has a carbohydrate component, and they're used in structures or the cell walls of plants. All right, cellulose does that. So here are the examples of carbohydrates: glucose, fructose, ribose, sucrose, lactose, maltose, and the other complex carbohydrates: starch, glycogen, cellulose, and chitin. Alright, and their example functions. Okay. Nucleic acids. So the two main nucleic acids we have are DNA and RNA. Alright, they're comprised of a 5-carbon pentose sugar, alright, which in the form of RNA is ribose and in DNA it is deoxyribose. They have a negatively charged phosphate group. Okay, this phosphate group bonds with the sugar, the pentose sugar, to create a phosphate sugar backbone. Alright, and it bonds through phosphodiester bonds. And there is also a nitrogen-containing base, all right? And there's 2 types of these, purines and pyrimidines, all right? Adenine, guanine, thymine, and cytosine. And of course, in RNA, T is replaced with U, so thymine is replaced with uracil. So nucleic acids, purines is a double ring structure, all right? Adenine and guanine. Pyrimidines are a single ring, okay? That's all there is to it. The nucleic acids run in the 5 to 3' direction. Okay, they have directionality. All right, the sugars bond with the phosphate. Okay, so the 5th carbon on the sugar molecule bonds with the phosphate, and the 3rd one bonds with the phosphate that is below it. All right, and then the bases come off the 1st carbon on the sugar molecules and point inwards. Okay, this allows them to create hydrogen bonds with the bases of the other strand of DNA. So, as I said before, DNA is made of 2 strands, okay? A, T, C, and G bond in the middle through hydrogen bonds. All right, now this is important to remember that A and T form 2 hydrogen bonds, C and G form 3 hydrogen bonds, and it's these hydrogen bonds that hold the DNA double helix together. All right. The 2 strands are known as being anti-parallel. This means they run in the opposite direction. So as you notice over here, the sugar molecule is pointing downwards. Over here, the sugar molecule is pointing upwards. They're running anti-parallel to one another. All right. Proteins. All right. The subunits of proteins are amino acids. Okay, and each amino acid has hydrogen atoms. It has a carboxyl group, which is just a functional group, an amine group at the other end, and an R group. So the R group is what gives the amino acid its unique properties. Okay, and it's distinguishable. All right, and these R groups can be either polar or non-polar. So when we look at protein structure, we look at 4 sequences. The first one being primary. Okay, primary is solely the sequence of amino acids. All right, the linear sequence of amino acids, ordered. All right. The secondary structure is when this linear sequence folds into alpha helixes and beta pleated sheets. All right. There's also random segments. All right, but these alpha helixes twist and beta pleated sheets are like sheets. All right, sit one on top of one another. Then, when those sheets, all right, beta pleated sheets and alpha helixes that are formed through hydrogen bonding come together, okay, they form the tertiary structure, which is a 3D conformational, fully functional structure of the protein, okay. This structure, the way it is folded, all right, determines the protein's biological functionality, so what it is intended to be used for, okay. This protein is to form properly in order to do its essential function that the body requires, all right. Now, often in some examples, proteins will fuse together to create something called a quaternary structure. Okay, this is when 2 or more polypeptides join together. An example of this is hemoglobin. All right, so in your red blood cells, you have molecules of hemoglobin, which are made up of 4 molecules, 4 protein components joined together to form the quaternary structure that is hemoglobin. So proteins can do structural functions, transportation functions, and regulatory functions, and these are some examples of them. So when we talk about making a biomacromolecule, we talk about synthesizing it inside the cell or digesting it. All right. Now, when you synthesize carbohydrates, proteins, and nucleic acids, all right, when the subunits form together, okay, to form polymers, all right, this is known as a condensation polymerization reaction. And what this means is the polymer that is made, every time a monomer joins together, it releases water. Condensations. Majority of biological reactions are known as condensation reactions. Lipids are not polymers, all right? They are composed of distinct groups, all right, and they do not undergo condensation reactions. So here's an example of a polymerization condensation reaction of two glucose monomers, all right? So when they join together, the two OH groups join together, and what this releases is water, all right? So they form together and they release water. Exactly the same when nucleic acids, when the backbone of DNA joins together, the sugar phosphate backbone, all right? When the OH groups bond together, all right, they release water when they fuse, all right? Hope you enjoyed it. Thanks for watching.